Hey everybody, welcome back. We're starting our next little segment here in Unit 4 about the muscular system. So in this video, we're going to be talking about excitation-contraction coupling. It sounds really complicated, but we're going to try to break it down so that we can understand on a molecular level what's happening in order to get your muscle cells to contract. All right, so the whole process here, I'm just going to give you this overview. So if I want to contract one of my muscles, one of my skeletal muscles, so one of the ones that I have control over, the first thing I do is have a thought in my brain, right? So my brain is going to tell me it's time to contract this muscle. That is going to send a signal down through my nervous system. So the signal, an electrical signal, is going to travel through my nervous system, and it's going to find its way to where the nervous system meets muscle cell. Where a nerve cell meets a muscle cell, that's called a neuromuscular junction. All right, and the electrical signal that's traveling down that nerve is called an action potential. So an action potential travels down a nerve, it finds its way to this neuromuscular junction, at which point that electrical signal needs to be converted to a chemical signal. And that chemical that gets actually travels across the gap between the neuron and the muscle cell is called a neurotransmitter. All right, so we have this electrical signal traveling down the nerve. It gets converted to a chemical signal in the form of a neurotransmitter. And that neurotransmitter is then going to trigger changes to happen on the surface of the muscle cell. So now this is the nerve cell talking to the muscle cell. Right? One of the big changes that's going to happen is a release of calcium ions inside that muscle cell. And that's going to cause their thick and thin filaments, remember that we talked about last time, so the myosin filaments are the thick ones, the actin filaments are the thin ones. It's going to cause that extra calcium in there is going to cause those two filaments to react with one another and cause the sarcomeres to shorten. And an overall shortening of the sarcomere is what results in the overall shortening of a muscle fiber, which gives you an overall shorter muscle, which is what we recognize as being a muscle contraction. All right, so a lot of steps happening there. We're going to kind of go through them one by one and make sure that we understand each step. All right, so here's a view of an actual micrograph view of a neuromuscular junction. So in this picture, we can see the branches of a motor neuron, so a part of your nervous system, reaching out and getting really, really close to individual skeletal muscle fibers. So you can see the skeletal muscle fibers in this picture. You can tell they're skeletal muscle fibers because of the striations, and we can see the little ends of the neuron reaching out and almost touching those muscle fibers, but not quite. All right, so here's a kind of drawing of what that same neuromuscular junction looks like. So here in this drawing, we can see that the axon terminal of the nerve gets really close to the muscle cell, but doesn't quite touch it. So the tiny little bit of space there is, let me grab my highlighter here. So the tiny little bit of space there is between the nerve cell and the muscle cell is called the synapse, right? So synapse is the space between the muscle cell and the nerve cell, right? So that gap is something that's going to need to be crossed by the neurotransmitter, right? So we can send the electrical signal or the action potential all the way down through the axon terminal of the nerve here, but when it hits the end of the cell, it has to be converted from an electrical signal into a chemical signal, and that chemical signal will be the neurotransmitter that crosses that synaptic cleft. All right, so here we can see that same process again here. So up here is the nerve cell. We have that action potential that's coming down the nerve cell as it reaches the end of the neuron here at the end of the axon. It's going to be converted into a chemical signal. And basically that chemical signal, the neurotransmitter that we're using for this particular purpose is called acetylcholine. So acetylcholine is our neurotransmitter of motor activity or muscle activity. Right, so as the action potential comes down that neuron, it's when it reaches the end of the neuron, it's going to convert from an electrical signal to a chemical signal by releasing these vesicles that are full of acetylcholine. So these vesicles then migrate down to the end of the axon terminal, we open them up. So through a process of exocytosis, we release the acetylcholine into the synapse, and that acetylcholine is going to travel across the synapse, and it's going to pick up on, or there are receptors on the muscle cell that are going to pick up that acetylcholine. Right. One more note on this slide is this term acetylcholinesterase. So this is an enzyme that just cleans up extra acetylcholine that's left over in the synapse. So after we send that acetylcholine across the synapse and it triggers the response on the postsynaptic cell there, then we need to clean up whatever's left over in that synapse, and that's the job of acetylcholinesterase. Okay. All right. So this concept of an action potential, we talked about it briefly when we were talking about just how muscle cells and nerve cells work in general, how they have to be excitable cells. Um, we also talked about this way back, maybe in week 1 when we were discussing transport, mechanisms of transport across the surface of the cell. We mentioned that some of your transporters, your cell transporters, are there to move ions from one side of the cell to another. Right, so, and this is, or one side of the cell membrane to another, I should say. This is going to be critical for the function of muscle cells and nerve cells. Right, so it's critical that these type of cells that are excitable are going to be maintaining a difference in charge on the inside versus the outside of the membrane. Right, so let's look first at this picture over here on the left side. All right, so this picture is showing, it's kind of zoomed in on a cell membrane when the membrane is at normal resting potential. So our resting membrane potential means that each one of your cells is going to kind of act like a little battery, where we've got a little bit more negative charge on the inside and a little bit more positive charge on the outside of that membrane. So we have a difference in charge across the surface of the membrane, right? So usually cells, when they're resting, have a resting membrane potential of negative 70 millivolts. So let me highlight that. So negative 70 millivolts is the voltage on the inside of the cell. Right, so if we're negative 70 on the inside, that means we actually have a positive charge on the outside of the cell, right? And you can imagine this kind of goes in conflict with what we talked about when we were discussing concepts of diffusion and how cells always want to kind of have this even charge, even concentration across a membrane. So we actually maintain this negative membrane potential through active transport. So if you can remember, the sodium potassium pumps that we talked about a couple weeks ago. So a sodium potassium pump is a protein pump that we find on the surface of your cells, and its whole job is to maintain this negative membrane potential, right? And it does so by moving sodium ions and potassium ions. Basically what it does is it ships three sodium ions outside of a cell and receives 2 potassium ions to the inside of the cell. And by doing that, it's pushing more positive ions outside than there are negative ions, or there are positive ions inside, which gives us an overall positive charge on the outside of the cell. Right, so these sodium potassium pumps are going to be really important for understanding why the cells are able to maintain that negative 70 millivolts of resting membrane potential. Okay, now moving to the inside of the cell now. So we're going to skip ahead for a second. We'll come back to that resting membrane potential, but let's skip ahead and talk about what's going on the inside of the cell that actually results in contraction of the muscle. So we talked about myosin and actin, the thick and thin filaments that are inside your myofibrils, which are inside your muscle fibers, and how these two filaments, these two proteins, have to interact with one another in order to shorten the length of the sarcomere. All right, so the thing that's actually shortening the length of the sarcomere is an action where myosin has these heads. So a myosin head right here. That myosin head is strongly attracted to actin. So here's our actin protein up here. So the myosin head really wants to attach to that actin and pull it. But it can only do so when given permission. All right, so it'll only contract when given permission, and we'll talk about what the permission looks like here in a second. All right, so when it has permission, what it'll do is if here's your actin filament and here's your myosin, the myosin will grab onto the actin and pull it. And if we're pulling from both ends of the sarcomere towards the M line, then overall we shorten the sarcomere, and that's how we get a muscle contraction. All right, so step one of this process, we have to allow the myosin to grab onto the actin. And the first step is actually to energize that myosin. So right here, we can see a myosin head, and here's our actin up here. Here's another myosin head. We can see here's an ADP plus an inorganic phosphate. So that's what happens when we use an ATP molecule. So ATP is your molecule of energy, right? That's your energy currency molecule. We actually use the ATP in terms of muscle contraction. We use the ATP in order to pull the myosin head back, right? So the myosin head at this point is being pulled away from the actin. So it's being pulled in this direction, kind of backwards away from the actin, right? And we're able to pull it away by giving it that ATP. So once the ATP has been used, it becomes ADP, or adenosine diphosphate, and plus an inorganic phosphate molecule there, right? So that's the first step of this process. That's called the ATP hydrolysis, because we're hydrolyzing ATP into ADP, right? And so that's basically pulling the myosin head back away from the actin. Now, I want you to focus in on what's going on around this actin. So the actin looks like these little yellow beads up here, but we see another couple protein molecules that are associated with that actin. Those other protein molecules up there are called troponin and tropomyosin. And troponin and tropomyosin are going to be important in kind of guarding actin or protecting him from myosin, right? So think about troponin and tropomyosin as being sort of chaperones between this relationship between actin and myosin. So myosin wants to grab onto actin, but troponin and tropomyosin are going to stand in the way and block myosin from grabbing onto actin until it's time. All right, so even though we hydrolyzed and we have the myosin head cocked backwards, ready to grab on, it can't grab onto the actin until troponin and tropomyosin move out of the way, right? So that's what's happened in this second picture here. So the troponin, which are these kind of purpley blue ones here, and the tropomyosin, which is this long stringy one, those guys are going to move out of the way, and that's going to open up some of these binding sites on actin. So there's some actin binding sites there that the myosin wants to grab onto. And once troponin and tropomyosin are moved out of the way, that means the myosin head can grab onto the binding sites on actin. And when they're connected like this, when they're connected together, we call that, they've formed a cross bridge. So a cross bridge is when there's this connection between the actin and the myosin. All right, next step of this process is called the power stroke. All right, so power stroke is when now the myosin has grabbed onto the actin. So here's our actin and here's our myosin. Now the myosin is going to pull, right? So it's going to grab onto that actin and pull the molecule sideways, right? So we have some myosins pulling sideways in this direction and some myosins pulling sideways in this direction. We overall get a shortening of the sarcomere as all those proteins begin to overlap. So the power stroke is when the myosin just pulls against the actin so that we can bring the Z lines closer together and shorten the sarcomere, right? And then the last step of this process is we have to detach the myosin from the actin. And the way that we detach it is just the way that we started this whole process. We have to pay a little bit of ATP. So we hydrolyze an ATP molecule, and that ATP is what is basically paying the myosin to reach back then and let go of the actin temporarily. Okay, so here's some images of what this looks like. So the black and white here are actual like microscopic images of what a sarcomere is going to look like at different stages of contraction, right? So, and we can see it over here. There's a lot going on in these pictures, but just focus on the Z lines. So here's a Z line here, here's a Z line here, and then right in the middle we've got, or here's another one here. So here's a Z line here, and then right in the middle we've got an M line. Right, so an M line is our middle line. So here we can see if a Z line is here and a Z line is here, we are totally relaxed. So this muscle is completely stretched out. It's not contracted at all. Here, we'll focus on just the right-hand side of this image. Here, the Z lines are getting a little bit closer together. So as we pull those Z lines a little bit closer together, we can see the actin, which is here, the yellow stringy things here, is sort of overlapping a little bit more with the myosin, and our overall sarcomere is getting shorter. This one down here is fully contracted. So we can see our Z lines are very close together, and we've got total overlap between the actin and myosin. So the myosin has completely pulled the actin in so that the Z lines are as close together as they possibly can be, and that's the shortest we'll be able to make that particular sarcomere. Right? So if we have that kind of shortening for every sarcomere over the entire length of a muscle, right? So just keep on shortening and shortening and shortening those sarcomeres, then eventually the entire muscle is shortened, and that's what allows you to do work. When you shorten a muscle, it's going to pull on the bone that it's attached to and cause that bone to move. Okay, I kind of like this image because it makes sense to me what the myosin is doing. So here the myosin heads are represented as these little blue men. Those little blue men, the myosin heads, are grabbing onto the actin, which is the pink filaments here. And that's what causes the muscle to contract. So as the myosin pulls on the actin, we're just going to pull those ends, those Z discs, closer together. That's what's going to overall shorten the sarcomere, which overall will shorten the muscle, which will lead to useful work being done by your muscles. Okay, so now that we kind of understand the basics of what's happening in terms of shortening the sarcomere, let's look back at the kind of chemical and ionic things that are happening in order to trigger this sort of contraction. Right? So remember that this all starts with a brain impulse. So your brain says, I want to move this muscle. It's going to send an action potential, an electrical signal down this axon here. When that action potential reaches the end of the axon, it's going to trigger these little vesicles down here. These vesicles are full of acetylcholine. Those vesicles are going to then undergo exocytosis. They're going to merge with the edge of the membrane and then push their contents out into the extracellular space. And their contents are acetylcholine. So the acetylcholine moves into the synapse, which is the gap between the nerve cell and the muscle cell. And that acetylcholine is going to be picked up by surface receptors here on the surface of the muscle cell. When we receive that trigger, when the muscle cell receives that message of acetylcholine, it's going to trigger a change in voltage. It's going to tell all of these sodium potassium pumps that instead of continuing to maintain that resting membrane potential of negative 70 millivolts on the inside and a positive charge on the outside, it's basically going to cause a whole lot of doorways in the cell membrane to open up. And when those doorways open up, the ions are going to want to equalize themselves across the cell. So once the doors are opened, it's really difficult to maintain your negative charge between one side to the other. So what will happen is you open a doorway, a whole bunch of sodium ions are going to flood into the cell, right? So the sodium ions are just going to start flooding into the cell. And because sodium is a positive ion, all of a sudden now our cell is not going to be negative on the inside anymore. Now it's actually going to be positive on the inside because we've let all these ions rush in, right? So now follow along here in this picture. So sodium ions are coming into the cell. That's giving us now an overall positive charge on the inside of the cell and a negative charge on the outside of the cell. The fact that we've reversed that membrane potential, so we went from negative on the inside to positive on the inside and positive on the outside to negative on the outside, that's an action potential. So an action potential is when you reverse the polarity, right? So we had two poles, a positive and negative, and then we reversed them, right? So that's an action potential. That message is going to continue to trigger the opening of these channels, these ion channels along the surface of the cell. So we open this channel and that triggers us to open this channel and then this channel and then this channel and that triggers the next one and the next one and the next one. It's just going to continue to trigger the opening of these channels, and that action potential is going to continue to move along the surface of the cell. All right, so as sodium ions rush in, that's our action potential. That creates a reversal in the polarity, right? What that does, and this is the next step now, so once we've received that action potential, it's going to trigger a release of calcium ions on the inside of the cell. So here is our sarcoplasmic reticulum. So sarcoplasmic reticulum is a muscle cell's version of the endoplasmic reticulum, or the smooth endoplasmic reticulum, the SER. Um, in this case, its whole job inside of a muscle cell, instead of having where normally smooth ER deals with synthesis and storage of lipids, in a muscle cell, the sarcoplasmic reticulum deals with storage of calcium ions. So we see we've got this big stockpile of calcium ions inside this sarcoplasmic reticulum. When we receive the message, when that action potential triggers this sarcoplasmic reticulum, it's going to release all of those calcium ions out of the sarcoplasmic reticulum, and they're going to go everywhere around the inside of the cell. So we're going to just see calcium ions kind of going all over the inside of the cell. And I'll show you in a minute how those calcium ions are going to be related to the actual contraction of the muscle cell. Okay. All right. So here's looking at this concept again. So we've got an action potential that's moving along the surface of the cell, so moving along the sarcolemma, which remember is the name of our muscle cell membrane. So we've got an action potential moving across the sarcolemma. As that action potential reaches the sarcoplasmic reticulum, it's going to cause calcium ions to be released. Those calcium ions are going to find their way all the way down to the inside of the myofibrils. And remember, on the inside of the myofibrils is where we have our myosin and actin filaments. So down here in this picture, we can see the troponin and tropomyosin, which are our chaperone proteins that are kind of surrounding actin and protecting it. When calcium gets in the way, though, calcium, think of it as like a distraction. So the troponin is going to be greatly distracted by the presence of calcium. So calcium is going to come up and it's going to distract troponin. Troponin then will change its shape because it's interacting with calcium. When troponin changes its shape, it moves tropomyosin out of the way. And moving tropomyosin out of the way leaves open binding sites on the surface of actin. Right, so now we've got these binding sites on the surface of actin. So our myosin heads can reach out and grab the actin. They weren't able to grab it before because the tropomyosin was in the way. But once we distracted the troponin with calcium, the troponin moved, which caused the tropomyosin to move, which opened up clean binding sites on actin so that the myosin heads could grab onto actin and initiate the power stroke. All right, so now that the binding sites are open, we've created that cross bridge, and then the myosin is free to pull on the actin and overall shorten the sarcomere. Okay. All right, so here's the same thing again. Just we're going to go through the whole process again. Hopefully it's going to start to sink in and make a little bit more sense the more I keep repeating myself here. All right, so starting at the top here, we've got an axon terminal, which is part of your nerve cell. We send an action potential, which is that electrical signal, down that axon terminal. When it hits the terminal bulb, which is the end of the terminal here, it's going to trigger the release of neurotransmitters in vesicles. So those little vesicles, those little bubbles that are inside the axon terminal are full of acetylcholine. When the action potential reaches the end, those vesicles are going to open up and release that acetylcholine into the synapse. So the synapse here being the gap between the neuron and the muscle cell. Right, so once the acetylcholine reaches the surface of the muscle cell, so it kind of migrates across that synaptic gap, it's going to be received by receptors on the muscle cell, and that's going to sort of disturb the muscle cell into knowing, hey, someone's poking us, something's happening, we're told to do something now. That's going to trigger a whole bunch of those ion channels to open. When the ion channels open, because we've got overall negative charge on the inside, a whole bunch of positive ions that are on the outside of the cell are going to rush inside to balance out that negative charge. Right, so once we've got all of those ions rushing into the cell, now we've created a whole nother action potential on the muscle cell. So our initial action potential was on the nerve cell. It went all the way to the end of the nerve cell. Then it had to jump in the form of a neurotransmitter to the muscle cell, and then the muscle cell picked up that action potential and continued to carry it on. Right, so that's how this action potential gets moved to the muscle cell. As that action potential moves along the surface of the muscle cell. Those changes in charge, the fact that we went from having a negative 70 millivolt charge on the inside of the muscle cell to now having a slightly positive charge, is going to trigger the sarcoplasmic reticulum to open up and release a whole bunch of those calcium ions. Calcium ions are going to find their way to the troponin, which is bound to the surface of the actin, the thin filament. As the troponin binds with those calcium ions, troponin will change shape. And as he changes shape, he's going to push tropomyosin out of the way. Now that our chaperones, troponin and tropomyosin, are distracted by calcium, that leaves open binding sites on actin, which means that the myosin heads can reach out and grab the actin and start to pull. And that grabbing and pulling is the formation of the cross bridge and the power stroke, which is actually shortening the overall sarcomere. Right, so that's what gives us a muscle contraction. So it takes a lot of work, obviously, but that's what results in the overall contraction of a muscle. Okay, so we're going to go through it one more time. So different picture here. You never know what pictures are going to resonate with different students. So looking at this one, we've got our axon. So the axon is reaching out to this muscle cell. We've got the axon terminal there. If we zoom in on that axon terminal bulb, we can see that we've got the vesicles of acetylcholine in here. As the action potential reaches the terminal bulb, it's going to trigger those vesicles of acetylcholine to undergo exocytosis and push that acetylcholine out into the synapse. Acetylcholine will hit some receptors on the surface of the muscle cell, and that's going to cause the muscle cell to be excited. When he's excited, he's going to open up a bunch of those ion channels. It's going to cause sodium ions to rush in, and as those sodium ions rush in, that's going to trigger the sarcoplasmic reticulum to release calcium. Calcium ions are going to bind to troponin. Troponin then will change shape, move out of the way. As he moves, he's going to carry tropomyosin with him, which is going to leave the open binding sites on the actin. The myosin will grab onto the actin, and we'll have the power stroke, which overall will shorten every single sarcomere. And as we shorten this sarcomere and the one next to it and the one next to it, then overall, eventually we get an overall shortening of the muscle. And that's what happens when you flex your muscle, right? You flex your muscle, it gets a little bit shorter, right? And as it gets shorter, it's going to pull on the bone that it's attached to. Right. So that's how we move a skeletal muscle. All right, so moving on to different sources of energy that your muscle cells use. So muscle cells are highly active cells. So every time we want to flex one of those muscle cells, we use a lot of ATP, right? Because we needed the ATP in order to unhook the myosin from the actin, right? So in a way, contraction is actually the resting stage of your muscles. If you want your muscles to relax, you have to pay a little bit of ATP in order to separate the myosin from the actin, right? So that's why, so one of the reasons why when people pass away and they go into rigor mortis, that is their muscles going into the least energy state, which is contracted, right? The muscles actually contract because they don't have any ATP left over to trigger that relaxation, right? So we obviously need a lot of ATP in order to keep this whole excitation contraction coupling thing going, right? We need ATP in order to maintain the negative resting potential so that we can get our action potentials. We need ATP to remove the myosin to hydrolyze, so we can hydrolyze that ATP and remove the myosin from the actin head. So ATP is critical for all of this muscle work to happen. You only have about a 3-second supply of ATP in your muscle cells at any given time. So that's not a whole lot, which means that, I could flex this muscle for about 1 to 3 seconds before it's going to get tired out, and I'm going to have to move on to my backup energy supply. So 3 seconds of ATP, and then I move on to a stored energy in the form of a molecule called creatine phosphate. So creatine phosphate is a slightly longer term storage of energy. You have about 12 seconds worth of creatine phosphate power in your muscle cells at any given time, right? And then you have to, beyond that, you just have to be performing cellular respiration. And cellular respiration, if you can remember, is the process by which your cells take glucose, take food, and break it down, get the energy out of that glucose and use that energy to form ATP, right? So we need to constantly be eating food, not constantly, but you need to have a food supply, an energy supply, and you need to have oxygen. So those are the two major components of cellular respiration that we're going to need in order to build more ATP continuously inside your muscle cells, right? So oxygen is a really important step, a really important part of that process. Every once in a while, it's possible to run out of oxygen. This is usually only happens when you're doing very strenuous anaerobic exercise. So if I ask you to run a sprint, if you're sprinting, you're not physically going to be able to get enough oxygen to your muscle cells in order for them to make enough ATP. So they're going to go into a backup supply, which is creating ATP without oxygen. And that process is called lactic acid fermentation, right? They would prefer not to do that because it can builds up a byproduct in the form of lactic acid. All right, so here's a close-up picture of what that creatine phosphate pathway looks like. So if we have a little bit of extra ATP, and remember ATP is a really short-term storage molecule, what your muscle will do as it's resting and it has a little bit of extra ATP, it'll combine it with a creatine molecule, and that's going to give us this creatine phosphate. And then when we need it, when we start to use that muscle and we need that energy, then we're going to use this enzyme, creatine kinase, and that's going to reconvert us back to just the normal creatine and back to the ATP that we can use for contraction of that muscle, right? So creatine phosphate storage gives about 15 seconds of energy, 12 to 15. Okay, now glycolysis. We've talked about the steps of cellular respiration before, so hopefully glycolysis rings a bell. If not, time to go back and review what glycolysis is. But the basics is that glycolysis is the first step in the breakdown of glucose, and glucose is your main energy sugar, right? So when we break down glucose, which is a 6-carbon hexagon-shaped sugar, we're going to break it down into 2 3-carbon molecules, right? So here's our glucose, our 6-carbon molecule. We're going to break it down into 2 3-carbon molecules. Those 3-carbon molecules are pyruvate, right? So in the process of breaking glucose down into those pyruvate molecules, we also get a little ATP out of that. So we've broken the molecule in half, and that's released some energy in the form of ATP. Now, if we have oxygen present, so if you're not sprinting, in most cases you're going to have oxygen present, and we're going to go along this aerobic pathway and complete the respiration process. If we don't have any oxygen, then we go into this fermentation pathway where we're going to create a little bit of ATP, but mostly we're going to be creating some lactic acid as a byproduct, and that's going to cause some muscle pain. All right, so here's the overall process of cellular respiration. So you should definitely know this equation. So let me run through it real quick. So right here, C6H12O6, that's glucose, right? So that's your glucose molecule, 6-carbon sugar. So we need that glucose, that's the food that you eat. We also need oxygen. So this is the entire reason why you need to breathe in oxygen, is for this pathway to happen. So when we combine the oxygen with that glucose, that's what's going to give us the ATP at the end, right? So here's the ATP that we're doing this whole cellular respiration activity for. We also get carbon dioxide as a byproduct. So byproduct means we don't really need that, and we're just going to exhale it. You breathe it off and you get rid of it. We also get a little bit of metabolic water out of this process, which doesn't amount to a whole bunch of water, but a little bit of water every day, which we keep because water is good. All right. So all of the food that you eat is eventually going to undergo this process. So a lot of it is just coming straight from glucose. Some of it has already been converted to pyruvic acid. We can get this energy from fatty acids as well. So it's not only carbohydrates that you get your energy from. When we combine that with oxygen inside your mitochondria, so here's a mitochondrion, that's when we get a whole bunch of ATP. Right, so the cellular respiration process, the part of this that uses oxygen, if we have oxygen present, gives you up to 36 ATP for every 1 glucose molecule. So for every one single molecule of glucose, you get 36 ATPs out of it, right? And ATP is, again, critically important for muscle cells. They're really, really active cells. And your muscle cells are going to have more mitochondria than any other cells in your body because they're just constantly doing a lot of work, right? And part of that work is moving your skeleton around, but another part of that work is just maintaining body heat, right? And so as we're doing this process, one of the other byproducts here is heat, right? And that's what keeps you warm. All right. All right, so here's this glycolysis process again. And this is what this initial part of the process is glycolysis. So we take in this glucose, we're going to break it down into two pyruvic acids. In that process, we get a little bit of ATP. Now, if we don't have oxygen, so if we do have oxygen, then we go into that whole aerobic respiration thing from the previous slide. But if we don't have oxygen, that's when that pyruvic acid, instead of going into the mitochondrion and being converted into a whole bunch of ATP, it just gets converted into lactic acid. So lactic acid is our byproduct, and that's what makes your muscles burn. So if you're sprinting and your muscles get to a point where they're just burning and you feel like you can't take any more steps, it's because you've got a buildup of lactic acid in those muscles because they weren't receiving enough oxygen in order to be continuously making ATP. So this lactic acid fermentation gives you a little bit of energy, a little bit of ATP, you know, maybe hopefully enough to finish the race or to run away from whatever's chasing you or whatever is going on in your life at that situation. But it's a little bit. It doesn't last forever, and it's only in your skeletal muscles that are capable of doing this without oxygen. All right, so overall skeletal muscle metabolism. Again, we always need glucose. So the sugars coming in from the food that you eat. So you eat food, the sugars that are in that food get absorbed across the surface of your small intestine. That sugar winds up in your bloodstream. We need to get that sugar into your cells using insulin. Once it goes into your cells, then we can start to break it down. So we'll break it down into pyruvate. That pyruvate, if we have oxygen, will go into the mitochondria, and then we'll continue to do all of the cellular respiration process until we get a whole bunch of ATP out the other side. If we don't have oxygen, then we go into the fermentation process and we end up with lactic acid on the other side. Now, lactic acid is toxic. It's an acid that you do not want to have in your muscles. And it takes oxygen to clear that lactic acid away. So when you do this, when you push your muscles to the limit where they have run out of oxygen and they're building up lactic acid, you put yourself into oxygen debt. And oxygen debt means that even after you stop doing that activity, you're going to still kind of be breathing heavily. And you're breathing heavily in order to clear that lactic acid out of your muscles. So your respiratory rate, your overall metabolic rate might be increased for 24 to 36 hours after a heavy workout. And that's because you've created an oxygen debt in your muscles that they have to repay. Okay. All right, so that concept is known as excess post-exercise oxygen consumption. And that means that even after you're done exercising, you're going to be consuming more oxygen. Your respiratory rate or your depth of respirations might increase for several hours afterwards, even at a non-noticeable level, in order to repay that oxygen debt that was created by pushing your muscles to their limits. Okay. All right, so in the state of homeostasis, muscle use of oxygen and nutrients is balanced by the production of manageable levels of waste products. Yeah, so these are the waste products that you're creating. CO2, which you breathe off, right? So that CO2 that's created in your muscle cells gets pushed out into your bloodstream. Your blood is then going to carry it up to your lungs, and then your lungs are just going to exhale it off. The heat is really important, right? So the heat is what maintains your body temperature. Right, so we need to maintain homeostasis body temperature by producing a lot of that heat. And it's one of the reasons why if you're cold, if you're having a hard time maintaining body temperature, you'll shiver. And that shivering is your muscles contracting to create more heat. And then lactic acid, we mentioned, is a byproduct that happens when your muscle cells have to undergo anaerobic respiration. So when they've run out of oxygen, they'll produce that lactic acid. Okay, let's talk a little bit about cardiac muscle metabolism just real quick because this is a little bit different than what your skeletal muscles do. So your skeletal muscles usually have more time to relax, right? Your cardiac muscle cells, they have to be constantly contracting, right? So your heart is beating, 60 to 100 times every minute for for as long as you're alive. For every minute of your life, it's going to be beating at that rate, which means those muscle cells don't get a break, right? So they have to be able to get a really rich supply of oxygen, right? So they need lots of oxygen so they can create lots of ATP so that they don't ever get tired, so that they don't ever have to go into that anaerobic respiration, right? So one of the ways your body achieves this is by the first blood that's oxygenated that comes back from your lungs, that's full of the most oxygen, goes directly to the heart muscle itself. So that heart muscle gets this fresh supply of oxygen directly from the lungs. And that's going to allow it hopefully to never get too tired, right? If your heart muscle does run low on oxygen, if it's not getting the oxygen supply it needs, it some of those muscle cells may start to go into glycolysis, or they may start to go into anaerobic fermentation. And as they do that, and if they create lactic acid in your heart, that's very dangerous. All right, so that's angina pectoris. That's that chest pain that's usually associated with cardiac problems, right? It's because your heart muscle cells are not getting enough oxygen. All right, so here's a close-up look at some cardiac muscle cells. So just to remember that they have a branching pattern instead of that more parallel pattern that skeletal muscle has. And we also have these intercalated discs. So here's an intercalated disc here, separating these two cardiac muscle cells. Here's one here, here's one here. So these are just the discs that are separating the two cardiac muscle cells from one another. So these cardiac muscle cells have these slight gaps in between them because we need to be able to carry an electrical signal across the entire heart, right? We want the heart to be kind of beating in unison. We don't want this muscle cell over here contracting and then this one and then this one. We want them to kind of all be working as a unit so that the heart can pump blood in this rhythmic way. So in order to kind of increase the rate at which the electrical signals travel across the muscle tissue in the heart, that's what these intercalated discs are for. So inside those intercalated discs, if we look at them up closely, we've got these spot desmosomes, which are junctions that are going to be holding together those cells very tightly. And then we've got gap junctions. Those are going to be helping to transmit the electrical signal from one heart muscle cell to the next heart muscle cell so that we can keep that electrical signal going and they can continue to contract in rhythm. Okay, so I know this was a lot. It's a pretty complicated stuff, so feel free to rewatch it and go back to any parts that you have troubles with. And definitely ask me if you've got any questions. So we'll go over some basic questions here real quick. What is the source of energy for muscle contraction? Which protein is responsible for the actual contraction? What neurotransmitter crosses the neuromuscular junction, the synapse? And what ion is important in the release of that neurotransmitter and muscle contraction? Okay, so the answer is there. The main source of energy for muscle contraction is ATP, right? So you need to have that ATP in order to get a muscle contraction. The protein that's responsible for the actual contraction, the one that's moving, is myosin. So myosin, the thick filament, is the one that's actually grabbing and pulling on the actin. The neurotransmitter that crosses the synapse is acetylcholine. So acetylcholine is the main neurotransmitter responsible for motor activity in your body. And then the ion that's important for both the release of the acetylcholine and for the actual contraction of the muscle cell is calcium. So we need calcium ions in order to trigger the release of acetylcholine across the synapse and also to bond to the troponin and move tropomyosin out of the way so that myosin can grab on to the actin. Okay, next set of questions here. What do muscles use as energy reserve? So we've run out of our 3 seconds of ready ATP. What do we do next? What carbohydrates are muscles using to generate energy? And then when your muscles are working hard, what nutrient are they limited for? So say we've got enough glucose, we've got enough food. What's the limiting factor here for muscle contraction? And then what waste product are we producing when your muscles are working overtime? Okay, so energy reserve is creatine phosphate. So that creatine phosphate gives us a slightly longer term storage of energy, so 12 to 15 seconds of extra energy usage with the creatine phosphate. And then after that, we have to start generating new energy. And we generate new ATP by breaking down glucose. So glucose is the sugar molecule that all of your muscle cells can use in order to generate new ATP. Right? When working hard, our muscles are limited for oxygen. So oxygen is the main thing that they're limited for. So usually you can find a way to get some glucose or even some fatty acids from somewhere in your body to break down to get the ATP. It's the oxygen that's in short supply, right? And so if we don't have enough of that oxygen, that's when you start to go into that fermentation side. And then what waste product is produced when muscles are working hard? They're generally producing carbon dioxide, right? So as we use oxygen to break down glucose to make ATP, our byproduct is carbon dioxide. If they're working extra hard, and we have to skip that aerobic pathway and go into the anaerobic pathway, then the byproduct would be lactic acid. All right, so that's all. Again, I know it was a lot of information. Let me know if you have any questions. We can work through it together. All right, see you guys on the next one.